Hi and welcome to this cloning video, which is for Biology Triple students only. Now, a clone is an individual produced asexually and is genetically identical to the parent. This video will go through a variety of ways animals and plants can be cloned. Due to plants' natural ability to reproduce by cloning, gardeners have for many years cloned their best plants by taking cuttings from the original parent plant, placing the cutting in some hormone rooting powder, the hormone is normally auxin, as well as some compost, and the plant will hopefully grow into a mature plant which is genetically identical to the original. A limitation of this method of cloning is that you can only really clone one plant from one cutting, so for your efforts you don't get much back. If rather than taking cuttings, a few cells, also known as a tissue, is scraped off the parent plant and again put into a growth medium with some hormones like auxin, these scraped cells will begin to divide by mitosis and differentiate to a range of tissue types that will grow further into little plantlets, which can be picked up carefully by some sterile tweezers, and the individual plantlets can be transferred into compost, where you will get many clones of the parent plant. This technique is widely used by commercial growers and is a way to preserve rare plant species using just one single specimen. However, it is very important to grow these tissue cultures under sterile conditions to avoid them becoming infected with disease. Due to animals generally relying on sexual reproduction to produce offspring, and the fact that once tissues have differentiated in animals, it can be permanent and irreversible, cloning animals is somewhat trickier than cloning plants, and it needs to be done before the cells have specialised, which is at the embryo stage. And an embryo is needed, which is why what I'm about to describe is called embryo cloning. And it is a technique used by livestock breeders to clone offspring of their best animals. To get an embryo, the prize cow is given fertility hormones so that she produces a lot of eggs. You then fertilise these eggs whilst in the womb, using sperm from the prize bull. Once formed, the embryo can be gently washed out of the womb, and you then have a ball of cells containing DNA, which hopefully is a good mix of the prize money-making cow's genes. Embryos can then be split into individual cells, and these cells naturally divide by mitosis to produce embryos themselves. Each embryo is then implanted into the womb of a surrogate cow, and in each womb the embryos develop into calves that when born are basically all identical twins. Despite being expensive, this process allows a good cow to produce up to 3 times more offspring than it would naturally during its lifetime. The only drawback here is that there is no guarantee that the embryo that you are cloning is worth it, as who knows what mix of genes you will end up with. Adult cell cloning, however, can overcome this. Here you take an amazing prize-winning animal like a sheep or a pet that you'd like to clone. Take any cell from their body. Inside the nucleus of that cell is a blueprint or the DNA to make that being. So take out the nucleus, which is diploid, it has a full set of chromosomes, and bin the rest of the cell it came in. Now you need a cell that is programmed to divide. An egg cell is an ideal candidate. It already has a nucleus, but with half the number of chromosomes, and we also refer to it as being haploid. An egg cell will only divide once it gets a full set of chromosomes and become diploid during fertilization. We don't want to mix genes here, we just want the blueprint of the prize winning sheep. So let's remove and discard the egg's haploid nucleus and use an electric shock to encourage the body cell's nucleus to go into the empty egg. Once this is achieved, the egg becomes diploid and automatically will start to divide by mitosis, forming an embryo that can then be implanted into a surrogate sheep's womb, where it will develop into a clone of the sheep whose body cell the nucleus came from. Now, this process is very expensive and does have a low success rate. Dolly the sheep is a famous clone that was produced using this technique, and it took hundreds of thousands of attempts to actually produce Dolly, and she aged a lot quicker than she should have done. So there are teething problems that still need to be ironed out. This technique is also used for therapeutic cloning, so rather than implant the cloned embryo into the womb of a surrogate, in therapeutic cloning you split the embryo apart into individual cells and encourage them to grow into new organs for transplant. Typical exam questions on this topic could involve looking at different images of cloning and stating what type of cloning it is. So the first one here is adult cell cloning. Below it is embryo cloning. Both of these are the cloning techniques used for animals. Up here you have tissue culture, which is a way of cloning plants. And underneath, just to confuse you, this is not cloning at all. It is in fact sexual reproduction, where meiosis is used to produce the gametes, the sperm and the egg, both of which are haploid. When they go together, they produce a diploid cell, which will then develop into an embryo by mitosis. Now, the body cell in adult cell cloning that is used originally was produced through sexual reproduction, and by asexual reproduction or mitosis, this one fertilized cell grew into a multicellular organism. Similarly, with embryo cloning, the embryo itself is produced sexually by a sperm and an egg fusing together. It is only when that embryo is split up into individual cells does it become asexual reproduction. So again, you might need to distinguish points in a diagram like this that are sexual or asexual, or show mitosis or meiosis. Other questions that you could get linked to this is to talk about the advantages and disadvantages of each type of cloning technique. I've already mentioned those previously. Or the overall disadvantage of cloning. Cloning generally is good because you can make copies of something that is successful, like a prize-winning animal, for example. However, with any clone, there is no variation. They are all genetically identical. And if a new disease suddenly came up, no individual would have any resistance to that disease. And that could mean that if the disease was powerful enough, it could wipe out all individuals, and that species could go extinct. So that's all for this video. Make sure you can describe the steps in each cloning procedure and recognise them as diagrams, and evaluate the good and the bad for each one.